Hi students, once again I am meeting you to continue the lesson that is human reproduction. Under this heading today first I have to talk about the twins. What are twins? When two offspring are produced in the same pregnancy, then they are called twins. When two offspring are produced in the same pregnancy, they are called twins. We have three different types of twins. Number one, monozygotic twins. These twins are called identical twins. Another name for this one, identical. They are also called as identical twins. What's the meaning for that one? Here only one egg and one sperm involved. They are fused together to form a diploid zygote. They undergo, so the zygote thus undergoes cleavage. At the end of first division, we have received two blastomeres. Normally, the blastomeres do not separate under normal conditions, under normal development. But here what is happening? The two blastomeres after first cleavage are separate. And each blastomere develops into an embryo. So we are getting two embryos, and these two embryos are developed into what we have the twins. Two offsprings and the same pregnancy. And normally, there is only one placenta for these two offsprings. Okay, so I am repeating once again, monozygotic twins, the name is given because one zygote gives rise to two individuals after first cleavage division, hence called monozygotic twins. These twins are also called identical twins because they are of the same sex. They are all alike, actually alike, and have the same genotype or genes. Okay, that's why they are called as identical. So same genotype or genes, then actually have what is called the same structural aspects. So same sex, same phenotype, same genes, and are also they are alike. Hence called identical twins. So they have the same phenotype, and also you see that one the same sex. Both are either male or female. There is no female one and there is no male one. Both are of the same sex. This is called monozygotic twins. Now I have to go for another one, dizygotic twins. Now the second type, dizygotic twins, also called as fraternal twins, also by name non-identical twins. Here there are two eggs and each one is fertilized by a sperm. So we have what is called double fertilization occurs. One fertilization, another fertilization. Two eggs are released by the ovary at the same time simultaneously. And each one is fertilized by a sperm. Now we are getting what is called zygote 1 and also zygote 2. So two separate fertilization process. It also occurs because of what is called double ovulation process. Two eggs are released either from the same ovary or from different ovaries. That is not the case. Anyway, they are produced actually by means of double ovulation. Two eggs are released. Each one is fertilized by the sperm. So we are getting zygote 1 and zygote 2. And each one develops into an embryo and then forming what is called the twins. So these are the two twins. Now here, the two twins may be of the same sex, either both male or both female, or of different sex. One is male, one is female. That depends on the nature of the fertilization by the sperm. In the case of first one, dizygotic twins or identical twins, the sex is of the same type. Both are same type, either male or female. Here, may be of the same sex or different sex. One male, one female, or both male or both female. That is why it's called as dizygotic twins, because of the formation of two zygotes and each one develops into an individual. Such twins are called non-identical twins or otherwise called fraternal twins. Fraternal twins, they are formed by double ovulation process, formation of actually two zygotes, and each one develops into an individual. That's about what we have. That is the second type of twins, dizygotic twins. Now the third type of twins, Siamese twins, always called actually conjoined twins or united. Okay, that is normally the names are different but same, Siamese twins. So in the case of Siamese twins, It is nothing but actually what we have identical twins. So that is normally there is one egg, one sperm, fertilization resulting in zygote. The zygote undergoes cleavage to form two cells, two blastomeres. In normal conditions, for example, in the case of identical twins, the two cells get separated, the blastomeres get separated, each one gives rise to a twin. That's why it's called as what's called monozygotic twins. The same condition here, but here the two cells, namely the blastomeres, after first cleavage, fails to divide. The two cells fails to divide. As a result, what is happening? Twins are born with some of their parts fused together, and such twins are called Siamese twins because the two individuals, what are called the co-twins, fused their body parts in some places. So that condition is called what is known as Siamese twins. Why is it called as Siamese twins? The first conjoined twins was normally born in Siam, the country. That is Siam, the old name of Thailand. The old name of Thailand. So that's why the twins are called Siamese twins. First child was born in Siam, the old name of Thailand. That's why the name is given Siamese twins. So these are all the three different types of twins. Normally, the two offsprings which are developed in the uterus during pregnancy. Okay, that's about what we have twins, monozygotic or identical, dizygotic or we have fraternal. And the last one, Siamese twins, they are nothing but identical twins, but some of their body parts fused at the time of birth. So during birth, the parts joined together or conjoined together. Hence the name conjoined twins, as in the case of conjoined vascular bundles, as we studied in the case of anatomy of vascular bundles. Okay, so with that I concluded about the types of twins. Now we have to go for the next one in the embryonic development. Now let's see the maintenance of pregnancy and the embryonic development. And before starting that one, after fertilization, zygote is formed. The zygote undergoes repeated mitotic divisions to form a hollow ball of cells called blastocyst. The blastocyst consists of an outer layer, what is called the trophoblast, single layer of large oval-shaped cells, and inner cell mass that later develops into an embryo. Now during further development, what will happen? The inner cell mass is differentiated into an outer epiblast, which later forms the ectoderm, and inner hypoblast, which later forms the endoderm. So the inner cell mass is differentiated into outer epiblast, which later forms the ectoderm, and inner hypoblast, which later forms the endoderm. In between the two, we have cells present, they together form the mesoderm. This is what is happening during development. Now gradually what will happen? The blastula gets converted into a gastrula. So the transformation of blastocyst into gastrula occurs. Now in the case of blastocyst, the early blastocyst, it cannot differentiate ectoderm, endoderm and mesoderm. During the embryonic development only we can get the ectoderm, endoderm and mesoderm layers. So what will happen? The blastocyst gets transformed into a gastrula with all the three germ layers, the primary germ layers, ectoderm, endoderm and mesoderm, by the movement of cells, and that process is called gastrulation. So after the formation of blastocyst, we have the next stage, gastrulation. That is nothing but the formation of gastrula. The blastocyst gets converted into or transformed into a three-layered gastrula with what we have the three germ layers, the primary germ layers, ectoderm, endoderm and mesoderm. That process is called gastrulation. So at the end of gastrulation we have formed the gastrula. Now the three germinal layers, the primary germinal layers, ectoderm, endoderm and mesoderm, They give rise to the tissues, organs and organ systems by a process called organogenesis. That's the next stage. You know, the gametogenesis, formation of gametes. Then we have the insemination process, the next stage. The next one, fertilization. The next one, cleavage. At the end of cleavage only we have formed the Blastocyst. And then we have the gastrulation process, formation of gastrula. With the 3 germinal layers, the layers give rise to the different organs and organ systems. Let's see later, by a process called organogenesis. That's about what we have, the pre-development. Now we have to go for the maintenance of pregnancy and embryonic development. And under this one, we have to study first extra-embryonic membranes. Before studying the pregnancy, first we have to know what are extra-embryonic membranes. The meaning for that one, those membranes present outside the embryo, not within the body of the embryo or within the body of the fetus. So those membranes present outside the body of the embryo are called extra-embryonic membranes. Now, based on the presence or absence of extra-embryonic membranes, we can classify the entire animal kingdom, particularly the vertebrates, not the entire animal kingdom. Specifically, the vertebrates are classified into amniotes and anamniotes. So, based on the presence or absence of what we have the extra-embryonic membranes, particularly the vertebrates are classified into amniotes and amniotes. For example, we have the classes reptiles, aves and mammals. These 3 classes are coming under the amniotes. The meaning for that one, they have the extra-embryonic membranes, one membrane called amnion, having all the membranes, but given importance for the amnion, one of the membranes, the animals are called amniotes with extra-embryonic membranes, reptiles, aves and mammals. Now anamniotes, we have what we have the paces and amphibians. Paces and amphibians, they do not have these extra-embryonic membranes during their development, hence called anamniotes. So based on the presence or absence of membranes, we have amniotes. Example, reptiles, aves and mammals with the extra-embryonic membranes. The anamniotes, example, the class paces and amphibians, all these are coming under the vertebrates. So here the extra-embryonic membranes are absent. Now we have to go for the functions of each one, etc. How far they are formed, either from ectoderm or endoderm or mesoderm. Now I will take the first one. So normally we have 4 different types of extra-embryonic membranes. 4 different types of extra-embryonic membranes. They are called amnion, chorion, that is yolk sac, then allantois. There are 4 membranes. These membranes are present outside the embryo, hence the name that is called extra-embryonic membranes. Now all these membranes are responsible for giving certain functions. What are the functions performed by the extra-embryonic membranes generally? So they protect the embryo from desiccation. Number 1, desiccation. The meaning for that one, protect the embryo from dryness. Number 2, mechanical shock. Mechanical shock. Number 3, absorption of nutrients. Absorption of nutrients through placenta. Number 4, just we have also exchange of gases. Exchange of gases. So the membranes all together, general functions I am telling, not specific functions, each one has its own specific functions. So amnion, chorion, yolk sac, allantois together perform certain functions. Number one, desiccation, preventing the dryness, actually protect the embryo from dryness. Mechanical shock, from the external shock, by providing a fluid medium around the embryo. Then absorption of nutrients through placenta, that's the nutrients are supplied to what is called the embryo. Then exchange of gases, oxygen is supplied to the embryo and carbon dioxide is removed. These are the four main functions. Now we have to take one by one the membranes. Amnion. So it is a double-layered translucent membrane. Double-layered translucent membrane, the amnion. Double-layered translucent membrane. Now I here represented what is called, this is amnion. I represented just one complete line and the dot. Together form double layer. So it is a double layer translucent membrane enclosing a cavity. The cavity is called amniotic cavity, filled with amniotic fluid. Now this is amnion, this is amniotic cavity. So the amnion is a double layer translucent membrane covering the embryo, having a cavity called amniotic cavity, filled with amniotic fluid secreted by this membrane, what we have amnion, and also by the embryo. So what is the function performed by this one? Now normally it provides environment, actually what's called buoyant environment. Buoyant environment. That is what's called the floating effect, buoyancy. It provides a buoyant environment to protect the embryo, that is a developing embryo, from what is called injury. So the shock is prevented. Buoyancy means the floating effect. So it provides a buoyant environment to the developing embryo from the injury. This is number one. Number two, it provides a medium for the movement of the sperm. It provides a medium for the movement of the sperm. That's the second function. Also, is having another function too. That is just keeping the embryo away from desiccation. And along with that one more, it regulates the temperature of the fetus. As per the book, only three functions are given. Number one, regulates the temperature of the fetus. Number two, provide buoyant environment to protect the embryo from the mechanical injury. And provides a medium for the movement of the embryo. Normally the embryo is surrounded by what we call amniotic fluid in later stage of development. So the fluid medium is always actually giving protection to the embryo from what is called the shock when compared to the solid medium. That's why the embryo is always surrounded by what we call fluid medium. So it is bathing in a fluid medium. That's nothing but the amniotic fluid present in the amniotic cavity. That's about the amnion. The next one I'll say what we call the yolk sac. So the yolk sac is nothing but a part of the gut of the embryo. See, this is the yolk sac. This is the yolk sac, what I mentioned here. So this is a yolk sac. It's a part of the gut. That is normally. It is concerned with the formation of early blood cells and early blood vessels. So a yolk sac is the second one which is not fully developed. The yolk sac is not fully developed. But somehow you have the yolk sac is present. Just normally it is connected to the gut of what is called, that's why I mentioned here, this is the embryonic gut. The shaded region is the embryonic gut, to which is connected what's called the yolk sac. It is concerned with the formation of early blood cells and early blood vessels. That's about the yolk sac. Now, the third one, allantois, I represented here. Actually, the allantois, this membrane is arising from what is called the caudal end of the gut. This is allantois. That membrane is normally actually arising from the caudal end of the gut. Caudal end of the gut means the gut of what is called embryo. Okay, and it is the base structure for the umbilical cord. It is a base structure for umbilical cord. So the umbilical cord is mainly formed from allantois. That's why it's called, it's a base structure that forms actually the umbilical cord, or the base structure of the umbilical cord. The one which connects the embryo to actually the placenta. So umbilical cord is the one which connects the embryo to the placenta. This is one. Second function, this allantois is also responsible for actually the formation of urinary bladder. That's why sometimes it is aptly called as a kidney of the early embryo. Kidney of the early embryo. So, what we have the allantois. The allantois is actually formed from the posterior end or the caudal end of the gut. It is a base structure of the umbilical cord. The umbilical cord is the one which connects the embryo to the placenta. And the second function, or that what is called allantois, it also actually takes part in the formation of urinary bladder. Hence called the kidney, the embryonic kidney. Hence called embryonic kidney. Now the next one, chorion. The chorion is the outermost membrane which is normally protective in function. It normally surrounds the amnion and also other membranes. You see, this is a chorion, outermost membrane surrounding other membranes. It surrounds the amnion and also other membranes and also the embryo. And also it takes part in the formation of placenta, a vascular connection between what we have, the embryo and the uterine wall of the mother, that's called placenta. So the placenta in the case of mammals, particularly humans, is formed of what is called chorionic placenta. Also sometimes called chorioallantoid placenta, erythrocrural placenta, hemochorial placenta. I will tell you later about that one, not necessary now. So the chorion takes part in the formation of placenta. How does it later? So chorion, the outermost membrane, covering the embryo and other membranes, taking part in the formation of placenta. Yolk sac is concerned with the formation of blood cells and what we have early blood vessels. Then we have the allantois is concerned with what is called the formation of umbilical cord and also for the formation of urinary bladder. Okay, that's about the extra embryonic membranes. Here I represented what we a simple picture not there in the book. Refer that one. Now chorion, I mentioned already. You see that one I mentioned, placental chorionic villi. Now what is happening during development? The trophoblast cells produce finger-like projections. These finger-like projections produced by the trophoblast, nothing but what is called the chorion. And that trophoblast is produced into many finger-like projections called chorionic villi. These are called the chorionic villi, which contains fetal blood vessels surrounded by what we have sinuses containing the maternal blood. Now normally the chorionic villi are bathing in a maternal blood. So that's why I'm saying that one, the trophoblast now produces many finger-like projections called chorionic villi, which normally contains the blood vessels of the fetus surrounded by sinuses, spaces filled with what is called the maternal blood. So the chorionic villi is normally bathing in the blood of a mother, the uterine wall of mother. There you have the spaces and sinuses containing the blood. I will proceed further now. Now let's see the development of the four extra embryonic membranes. Now amnion and chorion, they have developed from an outer mesoderm and inner ectoderm during development. They have outer, what is called the mesoderm and inner ectoderm. Now if you are taking Yolk Sac and allantois, they have developed from outer mesoderm and inner endoderm. So in all membranes, we have outer mesoderm. But in the case of amnion and chorion, we have inner ectoderm. Whereas in the case of Yolk Sac and allantois, we have inner endoderm. That's about the development. During development, they have formed from outer mesoderm, inner ectoderm, or outer mesoderm from inner endoderm. Now I mentioned about the trophoblast during development produces finger-like projections. These finger-like projections are called chorionic villi, which contains the blood vessels of the fetus, and such blood vessels are surrounded by sinuses. Sinuses means spaces containing maternal blood. So the villi are bathing in maternal blood. Now normally the connection between what we have the villi and the uterine wall together called as placenta. So the uterine wall as well as placenta together form what is called actually the placenta. Now the chorionic villi normally fit into the cavities, what are called crypts in the uterine wall. So both together combined to form a disc-shaped placenta. The placenta is normally acting as a transitory or temporary endocrine line during pregnancy. It connects the fetus to the uterine wall of the mother through umbilical cord. So it is an organ, the placenta is an organ which normally the nutritive or through which the nutritive, the respiratory and excretory functions are fulfilled. So it is concerned with the nutrition, excretion and respiration. Now the heart develops at about 4th week of pregnancy. Now it normally circulates the blood through the placenta, through the umbilical cord and through its own tissues. So the heart develops at about 4th week of pregnancy. It circulates the blood through the placenta, through what is called the umbilical cord, the one which is formed from what we have the allantois, and also through its what is called own tissues. That's about what we have the extra-embryonic membranes. I just represented separately. Now here the same one present within the uterus. So the fetus within the uterus. Now what is the difference between embryo and fetus? The embryo is the one which is developing without embryonic membranes. What we can say extra-embryonic membranes. Once the embryo has developed the extra-embryonic membrane surrounded by extra-embryonic membranes, then it's called the fetus. That's about what we have the development of extra-embryonic membranes. Now we have the formation of what is called the gastrula, what I mentioned, the 3 germinal layers. The next aspect is the organogenesis, the formation of different organs and organ systems. Now organogenesis, what is that process? The process of formation of tissues, organs and organ systems from the 3 germinal layers, namely ectoderm, endoderm and mesoderm, is called organogenesis. So the 3 primary germinal layers act as just what is called the primitive tissues from which the various organs are developed. Now each layer is specific in giving rise to a particular what is called the organ. Now what are the ectodermal derivatives? We have many, but in the book only limited what is called actually derivatives given. So I like to take only as per the book. So the ectodermal derivatives are nervous system, that is the central nervous system including the brain and spinal cord. Then the peripheral nervous system, those nerves leaving from the brain and spinal cord to other parts of the body, including sense organs also coming under this category, ectodermal derivative. Then the epidermis and its derivatives, that is the nail, the hair, the claws, the feathers in the case of birds, not in the case of humans. So epidermis and its derivatives, the various glands, hairs, nails, etc. Now also mammary gland. The mammary gland is nothing but a modified sweat gland. So these are all the ectodermal derivatives. What are the mesodermal derivatives? The middle layer. All the connective tissues present below the skin and also within the body cavity. Then the muscles. The muscles, skeletal muscles, smooth muscles, etc. Then the bones and cartilages, both being formed only from the mesoderm. And also the organs of the urinogenital system, a combination of what is called excretory system and the genital system. For example, the organs like kidney, the ureter, the gonads, they have been developed from what is called the mesoderm. Even the blood vessels, the blood also developed from the mesoderm only. Now the endoderm, the epithelial lining of the digestive tract, the respiratory tract. The lungs also developed only from the endoderm, though I have not mentioned here, only the epithelial lining of the digestive tract as well as what is called the respiratory tract. The digestive tract, otherwise called, that one gastrointestinal tract. And also the associated digestive glands like liver and pancreas, and also the endocrine glands present just nearer to what we have the digestive tract and alimentary canal. The thyroid gland and the parathyroid glands also develop only from what is called the endoderm. So these are all what we have the different types of actually the organogenesis, different types of what is called the organ formation from the three germinal layers. I listed only the organs. These are all the derivatives of ecto, endo and mesoderms. That process called organogenesis. With this I conclude. I want to take one more class to complete this lesson. That is what is called human reproduction. In the next class I will conclude. Okay, thank you.